Good evening to you all. And it's a very hiccup evening. So a lot of hiccups we had, but I think sure the talk will be so enlightening to you. So as part of COMBICS, the Student Association of Bioinformatics Department, so we are conducting a series of national webinars. And in that series, today's is the last session. Actually, in spite of all the troubles and troubleshootings that were done, and we are extremely sorry for all this delay, and hope the speaker will be understanding the technical difficulties that we have been facing here from this side. And it's one of the joyous occasion to introduce a speaker, a brief, extended introduction about the speaker will be done by the BTech student, Nivisha. But before that, it's my pleasure to invite once again the speaker, Dr. Vigneshwari Subramaniam. This is the second time I'm inviting her as speaker for COMBICS. We had her talk on proteochemometric modeling in 2020 on behalf of COMBICS. And so we had a very enlightening, since that was a COVID period. And even in spite of that, it was the beginning time, and she was in person here. And she not only enlightened the students with the talk, also she was giving some practical career guidances. So how to improve your language and your personality, and how to make yourself to be skilled, capacity builded enough to face the industry expectations. So in that series, we continually had many speakers, and one of the notable speakers was Dr. Vigneshwari Subramaniam. And today also, as a part of Silver Jubilee celebrations of our university, after our university has been declared as Deemed University, we are happy to have a part of celebration for Silver Jubilee. And in that extended mode, we have this national webinar series, and today's talk on the enhanced drug discovery in the industrial scenario with the AI-driven mode. So it's going to be a very pleasant talk to you. And so after long delays, so since I do not want to extend more, I just cut short my introduction and have a bright session with her. So now I invite Nivisha to make a brief introduction about the speaker. Thank you.
Good afternoon, everyone. It's my great pleasure to welcome you all to today's webinar, organized by SASTRA University as a part of National Webinar Series in Bioinformatics and Computational Biology. We are truly honored to host one of our most distinguished alumni, Dr. Vigneshwari Subramaniam. Dr. Vigneshwari began her academic journey right here at SASTRA University, graduating in 2008 with a degree in bioinformatics. Her remarkable journey from the classrooms of SASTRA to global leadership in pharmaceutical research is truly inspiring. She holds a PhD in computational drug discovery from the University of Helsinki and has developed deep expertise in molecular modeling, cheminformatics, and computational toxicology. She further expanded her academic and research excellence through postdoctoral work at Germany, where she contributed to cutting-edge computational method in drug development. Professionally, Dr. Vigneshwari held impactful role at LabCrop Drug Development and Fortrea, where she played a key role in advancing computational approaches to drug discovery. Currently, she serves as Associate Director at AstraZenca, Sweden, one of the world's leading biopharmaceutical company, where she continues to drive innovation in AI-driven drug safety and efficacy. Through her career, Dr. Vigneshwari has published in high impactful journals, led multidisciplinary international research collaborations, and played a pivotal role in applying technology to address real-world healthcare challenges. We are privileged to have her with us today to share her experiences and valuable insights. Please join me in extending a warm welcome to Dr. Vigneshwari Subramaniam.
Thank you so much for the introduction and for the opportunity. So maybe it'd be nice if you can mute from your end and I can start sharing my presentation. Is everything good? Can you see my screen?
Yes, ma'am. Yes, ma'am.
All right, great. Okay, great. Thanks a lot for this opportunity and for the kind introduction. I feel it's a great privilege to be back at SASTRA and speak again, even though I'm not physically there. So the focus of this talk would be more on to give you an overview of how drug discovery functions in industry and how do we make decisions while progressing compounds in the project as far as safety is concerned, because that's my field of expertise at the moment and that's what I would like to cover in this talk. So we already had a very good introduction, so I think I'll skip this slide. As everyone mentioned, yeah, it has been a long journey and I've been in Europe like for the past 15 years. And along with all the other things, apart from research, I've also been involved in a lot of mentoring programs. That's something I would like to highlight here. So I'm a mentor at New York Academy of Sciences, and I've also been mentoring high school students as a part of the Freedom Employability Academy in India. And along with that, I also mentor some PhD students and postdocs here elsewhere in Europe. And so if any of you need some kind of career guidance or if you just want to have a chat, feel free to reach out to me. So just to start with the very basics, so as most of you know, just because we design a molecule, it's not going to become a drug. So there are a lot of challenges associated with drug discovery. So it can be coming from the complex disease targets, or it can also be like the drug is staying for too long in the body. It can be coming from adverse drug reactions or poor absorption of the molecule in the body, or maybe it's just that it's highly unsafe. And also the commercialization potential and the competition in the market and the practicalities related to synthesis of the molecule. So all these things play a huge role in making a molecule actually becoming a medicine. So here the next few slides would focus on how can we actually use machine learning to support designing better molecules. So this is just a typical drug discovery setting. So once we start, okay, we start with understanding the disease. Let's say we are actually designing a molecule for cardiovascular diseases. Then of course we need to understand the disease. So it starts with target identification and then the hit identification phase. And then once we have the hit identification phase, again, we'll have like hundreds or even thousands of molecules. But then of course, at the end of the day, we just want only one drug molecule in the market. So then comes the lead identification and the lead optimization phase. So that's what we call as DMT, the design, make, test, and analyze cycle. So we design a molecule, we make it, we test it, we analyze it, and then this whole cycle gets repeated until we find that optimal drug candidate. And then once we have the drug candidate, then we go for the preclinical studies, like doing studies in rats, dogs, and mouse. And then comes the clinical trials from phase 1 to phase 3, and finally the drug is approved and launched. So as far as machine learning is concerned, it is actually supported a lot with decreasing the time taken for bringing a molecule to the market. So historically, it has been like 10 to 12 years to bring a molecule to the market. But lately, most of the companies have been using AI quite extensively, and this time frame has been reduced significantly. And also, the success rates of a molecule becoming a medicine has increased in the recent years. So as I mentioned before, so the whole drug design process, it's a multi-parameter optimization problem. So it's not only that like, okay, you have a molecule and it just becomes a medicine immediately. So you need to account for the ADME properties, that is the absorption, distribution, metabolism, and excretion of the drug molecule, and the efficacy of the molecule, commercialization potential. I mean, just because we have a molecule, we also need to see if it's even possible to synthesize that molecule and how safe is the molecule. So how do we do in industry is like we have a whole bunch of in silico models that supports with accelerating decision making. For example, in AstraZeneca, we've got about 26 models covering various properties. Like, I mean, for example, we can predict the various physical properties like solubility of a molecule, lipophilicity of a molecule, pKa, or the polar surface area. And And we've also got models to predict the in vitro ADME and in vivo ADME properties. When I mean in vitro, probably most of you on the call know what is in vitro and in vivo. Anyways, I'll still repeat it. So in vitro stands for like the experiments that are done in the laboratories with the cells. And in vivo is actually doing the experiments in a living organism like mouse, rat, and dog. And then, so we do have models to predict both in vitro and in vivo ADME properties. And then we also have a whole bunch of safety models that helps us to account for off-target pharmacology and organ-level safety risks. So these models are widely used in all projects, and they enable predictions at the point of design. So even before a molecule is synthesized, we can use these models to make predictions. So as far as AstraZeneca is concerned, these models are used quite extensively, as I mentioned before, and we process about 500 million molecules every month. So the advantage of these models is that they are integrated with the generative AI platforms. So for example, there's a platform called Reinvent. There's also a publication around it. So if you're interested, you can just go and take a look at it. So since these molecules are integrated with the generative AI platforms, so as I mentioned before, like we have a target and we are interested in designing molecules for that target. And like, okay, these are the kinds of compounds that be interesting for this purpose. Then you can tell your generative AI platform, I want a molecule with so-and-so lipophilicity properties with optimal PK profile. So that's really an advantage of these in silico models. Then, since I said before, I'll be focusing more on safety in this talk. So let's just get into the very basics. Why is safety actually a concern in drug discovery? So this study has been conducted in AstraZeneca before, like back in 2010. So they found out like most of the molecules that fail in the clinics mainly occur because of the safety related issues. And this is mostly related to the compound structure. So they also found out from this study, a molecule is highly likely to succeed in the clinics when we have high confidence in the preclinical safety. So what can we actually do to build more confidence in the preclinical safety? So as a part of these learnings, we have developed a whole bunch of in vitro assays and in silico models to address the various safety issues. So if you're interested in learning more about like what did they find in this study, there's a publication here. You can just go and take a look at it yourself. So based on those learnings back in 2010, AstraZeneca redesigned their R&D framework. So they call it as a 5R framework. So focusing on the right target, right tissue, right safety, right patient, and right commercialization potential. If you look at the plots on the right, so the study that was done between 2005 to 2010 and 2012 to 16, if we look at it, overall, the safety-related attrition has reduced significantly, and the success rates of the molecules being in the clinics also increased. So overall, at least in AstraZeneca, we found 5 times improvement in productivity after adopting this 5R framework. So then, I mean, as most of you know, every drug that you take, it has got some kind of dosage associated with it. So it can be 10 milligrams, it can be 100 milligrams, or it can be 1,000 milligrams. So, of course, dose is the most important thing that plays a role in safety. If we look at the molecules that are withdrawn from the market, the 2 pie charts that you see here, majority of those molecules, nearly more than 80% of the molecules, they were administered with a dose more than 100 milligram, and that's what actually lead to safety concerns. So if we look at it, so of course, this kind of implies low dose leads to less risk of adverse drug reactions. If that's really the message, should we actually design a low dose compound? Because it's always about like if you have a high therapeutic concentration, that's always associated with toxicity. But then comes the part, also in the introduction, this was mentioned, and also I mentioned before. So it's not only that you're interested in delivering a safe molecule to the clinic, but you also want a drug that actually works. If you have a cardiovascular disease, so the drug should be made in such a way that it actually addresses the underlying cause. So that's when efficacy is really important. So what we usually do is we aim to establish some kind of balance between safety and efficacy while designing a new molecule. So there, I would like to introduce a concept called therapeutic index here. So, which is nothing but the ratio of, like, when you take a drug molecule, you have some kind of exposure in your body. So what is the highest exposure with no toxicity to the exposure with the desired efficacy? So that's what therapeutic index means. So most of the drugs that are currently in the market, they have a threefold therapeutic index in humans. And just to give you a flavor of what actually influences toxicity, sometimes it can just be the primary pharmacology. Let's say you're designing a drug for a GPCR target, maybe, but it has got a very high affinity to GPCR, then that can as such cause toxicity. It can be the primary pharmacology. Sometimes it can be the secondary pharmacology, like the off-target effects. Instead of binding to GPCR, if it binds to a whole bunch of kinases, then of course it's expected to cause a lot of side effects. And sometimes it can just be the chemical structure. The structure might have the potential to generate reactive metabolites. And in other cases, in majority of the cases, it's actually the physicochemical properties of the molecule. So if a molecule is highly lipophilic, then it's highly associated with various safety concerns like phospholipidosis and QT interval prolongation. Don't worry about these terms. I'll have much more details in my later slides. So how are we internally using these predictive tools to facilitate designing safer drugs? So we have got models to predict off-target pharmacology, for example, kinases. So internally, I mean, all this is like being pharma industry, we have a lot of internal data. So we do have the bioactivity or the binding affinity data for 400,000 compounds and 171 kinases. So overall, we're talking about 1.5 million data points. So what we intend to achieve here is, like, we have one model that's a multitask graph convolutional neural network model that can be used to predict the activity classes of 170 kinases. So you can do this simultaneously. And so these models, like, I mean, with the amount of data that we have fed into these models, they have very good performances. And chemistry often uses these models, especially the promiscuity scores, which is nothing but if you have a compound, how many kinases are hit by this compound. So that's what we call as the promiscuity score. So these scores are usually used to prioritize compounds in projects. And also sometimes we use these models for early selection of clusters when we do high throughput screening. An advantage of using a multitask model when compared to building 171 single task models is that, so for example, I mean, even though we do have a lot of data, but this is not a complete matrix. We don't have the data for all 171 kinases and for all 400,000 compounds. So this is not a complete matrix. We do have a lot of missing values. For some kinases, we have data only for maybe 10 compounds, and for some kinases, we have data for 100,000 compounds. So when we do this in a multitask setting, the data-rich kinases, they support the predictions of the data-deficient kinases. And then comes the next one, yet another safety assessment that we often do is phospholipidosis. This is nothing but the accumulation of phospholipids in the various organs. This is quite frequently observed in inhaled compounds. So there will be a lot of accumulation of phospholipids in lungs, and that's not really good because long-term accumulation can lead to permanent organ damage, and this is not something we want. So we also do some kind of safety assessments related to phospholipidosis while we progress compounds in the pipeline. So we have a couple of ways to do this. So we've got conformal classification support vector emission models. So when I mean conformal classification, it's not only that these models help to predict if a compound has a phospholipidosis status that is positive or negative, but these models also provide some kind of confidence. How likely is phospholipidosis to happen? So that's the advantage of these models. And along with the machine learning models, we also have some kind of physicochemical property-based analysis. For example, if the lipophilicity is about 1 or if the pKa base value is about 8, then we also conclude that it has got a potential for phospholipidosis. So what it means for the projects is that, like, I mean, they take into account both the physicochemical analysis and also the machine learning models to make decisions. And a little bit more on the organ level toxicity predictions and what we do here. For example, sometimes the molecules that are designed can actually cause drug-induced liver injury. So we want to make sure a molecule that's delivered to the market doesn't cause any liver injury or cardiac toxicity. So we do have a Bayesian model. So Bayesian model is nothing but a probabilistic model. Rather than giving you point predictions, it shows you how likely is this compound to cause drug-induced liver injury. So we have a whole bunch of in vitro assays that goes as inputs to these models. And these in vitro assays, they account for cytotoxicity, mitochondrial toxicity, lipophilicity, and we also feed in maximum exposure and the potential of a compound to generate reactive metabolites. That's what we call bioactivation. And then once the compound is fed in with all these assay inputs, we can predict how likely is this compound prone to drug-induced liver injury. And again, this is a publication from AstraZeneca. If you want to know more details about the model, you can just go and take a look at it yourself. And as far as the organ level toxicity predictions are concerned, the next one comes the QT interval prolongation. Most of you might have seen this kind of ECG waveforms before. So for example, here you see PQRST waveforms, and this is from an ECG. So if a compound is likely to cause QT interval prolongation, what it means is that there will be extended interval between the Q and the T waveforms, like you see here. This is a normal ECG, and this is a long, prolonged QT. So this is mainly coming from the increased interval between the contraction and relaxation of the muscles. So what it means is that it can increase the chance of cardiac arrest by 5 to 7%. So we really want to make sure that the compound that we bring to the market is not causing QT interval prolongation. Again, we have very limited data for modeling this kind of endpoints. So it's just a Gaussian process model that relies on the hERG inhibition. hERG is an ion channel. Quite often, like if a compound is likely to hit hERG, so the projects stay very cautiously. The reason being it can cause cardiac-related issues. So here actually this model is just based on 65 drugs, and we feed in hERG inhibition and maximum exposure as inputs to this model. And as an outcome, what we can see is how likely it is to have QT interval prolongation. So there are like two lines here, the green and the red line. And the green line it's a safe compound, and anything goes above red, that's when it's likely to cause QT interval prolongation. And in the x-axis, you see the maximum exposure. So the maximum exposure is like, if you take a drug molecule, what is the concentration of the molecule that remains in your body? So for example, here, if you look at it, if it goes above 100 micromolar, then it's likely to cause QT interval prolongation. That's when one should stay cautious and make sure that the maximum exposure should be below that. So how do we use these models is to flag early safety risks before we generate in vivo data. And along with all these kinds of in vitro assays and organ level safety risk assessments, we also have other approaches that are complementary to the machine learning models relying on chemical structures. So there's an approach, maybe some of you have already heard about it. There's something called cell painting. It's a high throughput imaging technique that's usually used for compound scoring, and it can also be used for safety predictions. So what happens in cell painting is that you get an image of the cell and you see how the features of the cell, how the cellular components vary when a drug molecule is actually administered to that cell. And so that was just a brief overview of all kinds of safety stuffs that we do in industry. And now a little bit more on how can we use AI to facilitate the in vitro or in vivo to clinical translation. Because at the end of the day, whatever experiments we do in vitro or in vivo, it doesn't matter. We should be able to translate this information to the clinic. So having said that, so a little bit more on how do we predict the pharmacokinetics of the preclinical species like rat, dog, and mouse. So we have a whole bunch of internal data. So we use the chemical structures along with the in vitro properties and the ADME predictions. So as I said before, so wherever we can measure in vitro properties, we use that, but it's not a complete matrix. So if you're missing some in vitro experimental values, then we complement them with the predictions from the in silico models I described earlier. So at the end of the day, we build a multitask model to predict the clearance, half-life, exposure, and volume of distribution in rats, dog, and mouse. So again, there are publications related to this. For more details about the model, you can just go and take a look at it. And along with the rat, dog, and mouse PK models, we also have models to predict the human pharmacokinetics. And these models are highly relevant because they guide compound prioritization. And this is just a simple random forest model, but what goes in as input is that along with the physicochemical and the in vitro properties, we also feed in a lot of in vivo properties. For example, since in the previous slide, you saw that we can predict in vivo PK, we can predict the pharmacokinetics in rat, dog, and mouse. So this can, since we can predict those properties, this can also be fed as input to predict the human pharmacokinetic endpoints like maximum exposure, volume of distribution. An advantage of having this model is that you can have a simulation or you can make an extrapolation. If this molecule is administered to a patient in the clinic, what really happens to that patient? Of course, it's not going to be 100% accurate, but at least you can have some kind of simulations or expectations of that molecule. So these models, they guide individual project decision making, and they are also integrated as a part of the de novo generative models. So one could even design compounds with improved pharmacokinetic profiles. So yeah, that's the overall summary about the clinical translation. And so now a little bit more of like, okay, so to summarize, okay, how can actually machine learning models support compound design? So most of the models that I described earlier, these are integrated into the augmented drug design platforms that we use internally. And the main goal here is to improve the speed and efficiency of the DMTA cycles. And these models, they help design compounds with optimal safety and PK profiles, and this can be done early on in drug discovery. And as I said before, we have the option to model human PK, and this kind of gives us very high confidence in the preclinical translation, from preclinical translation to patients. So I've been asked in several forums if artificial intelligence is going to replace scientists. I would say no. I would say maybe those who don't believe in the power of AI would be replaced by those who do. And all the models and everything that I described here, we have got contributions from multiple people in AstraZeneca, and I would like to acknowledge all of them here. And thanks to you for your attention and for your time. So now I would be happy to take up any questions. Thank you. All right.
That was a very insightful presentation. Thank you very much, very much. So we have got up which community you had been once a part in the journey with Sastra in 2008. And I think your presentation was very, very clear and up to the level of the students to catch up and follow. And I would like to invite the students, if you have any clarifications or discussions to have with the speaker. I'd like to yes, if anyone is wishing, I think we would like to invite you to come to the stage. We have, or else in the front, you can come and talk. There is a mic in the front. Whoever, I think there is a person who has raised his hand. Devadarshini, maybe online. She is present.
So you can read the questions, you can read chat.
You can open the chat and read.
Chat. What about ethical consideration of ML in drug discovery and reproducibility? I think that is the first question.
Great, I can try to address this first. Maybe you can try to mute your microphones from your end, because I don't know, for some reason I hear the echo. So can you mute the microphones in the room, please? Yeah, thank you. All right, so getting to the ethical consideration. So yeah, we do address this, and quite often we have committees internally who deal with the ethical concerns related to AI, and of course, that's the most important thing. And even though, like, I mean, now that we are using AI in every stage, so we are actually trying to address this, and we have internal committees to do that. Of course, that's really, really important, and we account for that in AstraZeneca. And as far as reproducibility is concerned, to the most, whatever we do, actually, we make sure that it's easily reproducible. But of course, in case of some assays, like, I mean, as far as machine learning is concerned, yeah, we can try to introduce reproducibility as much as possible. But then as far as the experimental data is concerned, so that's going to be quite a challenge in terms of reproducibility. But as far as machine learning is concerned, we do our best to have reproducibility in whatever we do. But there are some, I mean, concerns like overall in industry that some studies are not reproducible. And also there are quite a few publications around it where people have said, like, if you want to make something reproducible, what should you actually do in terms of model building and model validation? So this is also this is a very interesting question. This is also one of the topics that's being picked up in the industry right now. Did that answer your question, Sasikala, or do you need further clarifications? Okay, great. Thank you. And Sagip has a question about, we have generated model to design de novo drugs, but for clinical trial in vitro and in vivo, or, I mean, okay, you see that it's not aligning very well, and what's your suggestion? Yeah, I agree with you that it's not like, okay, whatever you do in vivo, it's not going to align 100% with what's going to happen in the clinics. So yeah, we do our best, and we try to, I mean, make sure that we bring the optimal molecule to the clinic. But then still there can be some challenges. Maybe whatever you observed in vitro and in vivo, perhaps it's not happening in the clinics. And this is an industry-wide problem, and everyone is actually trying to address it. But to the most, I mean, imagine, like, when compared to 10 years back, of course, the success rates have increased significantly now. We no longer have similar challenges, but still we can't do 100% in vitro to in vivo to clinical translation yet. We still have some challenges there, and everyone is working on it, but we are currently doing our best to make it possible. So earlier, like maybe a few years back, probably it was not possible to predict organ level toxicity or even human PK, but we have really gone to that stage now, so we can extrapolate what can happen in the clinics. But of course, we still have some gaps, and everyone is working on it to address that. Do we have, was that okay with you, Sagib? Great, thank you. And do we have other questions? You can either post in the chat, or you can unmute your microphones and ask. Okay, Anisha has got a question around how you analyze how much drug is absorbed by the humans in vivo. So, I mean, of course, we can't actually do experiments on humans before they go to the clinics. But the internal analysis, what we do is, so we do check for the in vivo properties in the preclinical species. We check in rat, dog, and mouse, and we see what is the bioavailability of this compound, because the bioavailability is going to relate to drug absorption. So we check for the bioavailability of rat, dog, and mouse, and then we also have the human PK models that has the potential to predict maximum exposure and bioavailability. So we gain some insights from these models and from these in vivo experiments to see what can really happen in the clinic, but we can't be 100% sure if we would have the same absorption in the clinics. Yeah, and I hope that was okay, Anisha. And Devadarshini, I see your hand up. Either you can unmute and ask, or you can post it in the chat.
Yeah, Devadarshini, can you come up once again?
Yeah, she essentially posted in the chat. She's got a question regarding the integration of real-world clinical data into AI models for safety prediction from your experience in industry. And how do you ensure that the models remain both accurate and generalized across diverse patient populations? That's a very interesting question, Devadarshini. So, I mean, let me answer the first part of the question. How are we integrating real world data? So, I shared a couple of models regarding liver and cardiac toxicity. So, this is actually relying on the clinical data. So, these are all FDA approved drugs and they are available in the market. So, this is real world data, and we are integrating the real world data in the clinics to develop these models so that we can gain some insights internally how likely is a compound to have cause liver injury or cardiac toxicity. And getting to the second part of your question, how do you ensure that the models remain both accurate and generalized across diverse patient populations? So I can't comment on the patient population because that's not something I do, because there are other teams in AstraZeneca who are doing it, so I don't want to comment on it without having all the details. But I can tell you how good are the models internally. I mean, of course, we do our best to do all possible kinds of validations, and we constantly update the model with internal data. So we make sure that it's both accurate and generalizable. But often, as far as the safety endpoints are concerned, the key challenges with the data, we don't have a lot of clinical data for our internal compounds. So that's where the key challenge is. I mean, we do our best to generalize as much as possible, but sometimes we do have some challenges in this respect. And hope that was okay, Devadarshini. And Srimati has a question. Is there any drug available in the market or in the clinical phase discovered through machine learning? So there are so there was this recent company called, I don't remember the name of the company. There are a lot of these AI companies. So at least they claimed there is a publication around it that they discovered a molecule through machine learning. But I don't think this molecule is in the market yet, because even though you design a molecule through machine learning, you need to go through the process, you need to do the in vivo testing, and you need to take it through clinical trials before a molecule is being delivered to the market. So even though they have shortened the phase of drug discovery through machine learning, so as far as like bringing a drug to the market, I think it's still an ongoing process. Even though they have discovered through ML, this drug is not in the market yet. Anything else?
Kindly bear with us. We are switching on for the open mode so that if there is no more questions, then we can wait. And the students, it has been turned on that you can speak to the microphone directly also. You need not wait for messaging or texting her if you have any discussions or queries to do. Yeah, Dr. Ashok sir has posted a question. Could you address that?
I can try to address that. So, which are the organs are of interest for organ-specific toxicity apart from liver and cardiac toxicity, and are machine learning models available for them? So the other organs that be interesting, at least FDA has recently released a kidney toxicity dataset. So I would say that's something that could be interesting. And of course, lung tox is also one of the most important organs. I mean, especially for inhaled compounds, we need to account for it. But there are not so many machine learning models related to lung and kidney tox. I think for kidney tox, there's one of the models that's available publicly. But there are a lot of them related to liver and cardiac toxicities. The reason being we have plenty of data for these two organs as against the other organs. So that's why the machine learning models for other organs aren't that popular. And the next question is, what are the dataset size considerations for different problems based on the models being created? So, I mean, since we are modeling clinical endpoints, these dataset sizes are quite restricted. So whatever drugs are available in the market are actually fed into these models. So if we think about like all the drugs in the market, it's more about like 2,000 plus drugs in the market. So usually if we model liver or cardiac toxicities, that's the maximum data set size we can expect. Of course, we can't do fancy deep learning stuffs on this. It has to be a simple or classical machine learning model for these kinds of problems. Hope that answered your question, Ashok. And then Anisha has one more question. When we integrated drugs via GA and base methods, sometimes the structures are more complex. How you synthesize this in the lab? Okay, yes. So there are also models to predict synthetic feasibility. At least we do have something internally. Maybe I'm not sure if that's published. So one could actually, yeah, I mean, quite often like generative AI models, they can generate really fancy molecules, and then like you have challenges with synthetic feasibility. But at least internally, we also integrate the synthetic feasibility assessments as a part of the generative AI models. So while generating a molecule with optimal properties, these models also account for the synthetic feasibility. And nowadays, you might be seeing a lot of papers coming around like retrosynthetic predictions. So we do account for that as well while generating molecules via generative AI models. Okay, great. Anything more from anyone?
So I hope that the questions have been well answered and clear. Is there anything else that you want to get clarified? It was very wonderful when we had her in person here on the same podium. She was here and she was sharing her motivational talk on employability also. So I think in 2020, 5 years back, the students were very much benefited by that. So they learned the skills. So similarly, also I think one or two questions in that line. Would you like to take, Dr. Vigneshwari?
Sure, I would be happy to take up if you have anything related to career guidance. Yeah.
So now it's open to you.
Have it.
A specific way if you want to get skill.
Hi, Vigneshwari. Suma here.
Yes, Suma.
Yes. I want to ask a question related to that. Ma'am was mentioning about career guidance. No, so how the students need to be prepared to accept the AI revolution in the bioinformatics field?
All right. So I would say actually keep yourself up to date with the latest technologies. So just because you were doing something 5 years back, it doesn't mean you have to do the same thing again. So that would be my advice here, because I mean, at least when I started my PhD in Finland, so it has always been around classical machine learning approaches. But now we are shifting more towards deep learning, large language models, and this kind of stuff. So as the technology is evolving, keep yourself up to date and be adaptable to to accept those changes. Otherwise, it's going to be quite a bit of a challenge because everything is evolving so rapidly before you could even pick up things. So keep yourself up to date with the latest technologies and also read a lot of papers, see what's actually happening in the field. And also, expand your network, talk with other people like doing AI in other areas. For example, like often have conversations with people doing AI in Volvo, for example. It's automobile industry, but still they do have similar challenges. So what I would suggest is like try to talk with people who are using AI in other industries. So this in fact helps you to gain better insights. Perhaps you can from that you can whatever you get from those conversations, this can in fact be implemented in drug discovery. So just keep yourself open to different kinds of things. So hope that answered your question, Suma. Do we have anything more from anyone?
Dear students. Right. So, Dr. Suma? Yes, to proceed? Right.
Yeah. So we can wind up, right?
Yeah. Right. So, now the latest trending scenario in the market of drug discovery, right? So, and we thank Dr. Vigneshwari Subramaniam for her valuable time and kind patience when we were facing the hiccups, and also answering the questions in detailed and very clear manner. So that it was so, I believe the present set of batches will be impressed and inspired by your career achievements and today's talk. Thank you so much. And now I would like to invite Mr. Gauri Shankar to present the vote of thanks. Thank you.
Good evening, everyone. As we draw this enriching webinar series to a close, it is my honor and privilege to deliver the vote of thanks on behalf of the organizing team. First and foremost, I extend my heartfelt gratitude to today's esteemed speaker, Dr. Vigneshwari Subramaniam, for her impactful session. Ma'am, your expertise and insights have truly broadened our understanding on the transformative role of AI in pharmaceutical safety and innovation. Thank you, ma'am. As this is the final session of the series, I would also like to take a moment to express our sincere appreciation to the distinguished speakers who joined us in the previous sessions. Professor Karthik Ramanan, Dr. Shamir Kader, and Professor Gautam Menon. Thank you all for making the series a rich learning experience. We are equally grateful to Sastra University's management, our dean, associate deans, and all the professors of SCBT for their unwavering support throughout this event. Special thanks to the Dean of SOC and Mr. Santosh for facilitating the audience video recording. A sincere thanks to our moderator, Dr. Satya, for guiding the session with such clarity and grace. A big shout out to the amazing Comics organizing team for their tireless efforts behind the scenes. Your teamwork made this session seamless and impactful. We would also like to thank the auditorium management and staff, Mr. Shivakumar, for their cooperation and support in making the in-person arrangements smooth and efficient. Finally, to all our participants, thank you for your enthusiasm, thoughtful questions, and active engagement. As we wrap up, we hope the series has sparked curiosity, inspired innovations, and created lasting academic connections. Thank you all once again. Have a great evening.
Thank you. Thank you so much for the opportunity, and thanks to you for all the interesting questions. Thank you. Have a good day. Bye.